The possibilities of neurotechnology are almost limitless. Neuromodulation treatments, such as deep brain stimulation or MR-guided focused ultrasound, are the most efficacious treatment methods for Parkinson's disease and some other movement disorders. These techniques allow us to target almost any part of the brain, but the trick is to know which part of the brain should we target with each disorder. Could we use these highly efficacious targets for any brain disorder? That's the aim of the work we do in Turku Brain Lab. Modern neuroimaging tools have revolutionized our understanding of brain functions and malfunctions. However, most data comes from case control studies, which are limited in terms of causality. We can characterize the abnormalities in the brain in any disorder, but the challenge is to know what changes actually cause the symptoms in the patients. Brain lesions, however, have this causal inference. When a patient with new onset patient suffers a brain lesion and develops new onset symptoms, then there's a strong causal link from that lesion to the symptoms. Now, if all cases where a brain lesion has caused similar symptoms hit the same spot in the brain, then we can be fairly confident that that spot in the brain is critical for the development of symptoms. However, the clinical reality is that lesions tend to hit different regions in the brain, even though they cause identical symptoms. At Turku Brain Lab, we believe that the solution to the localization problem is brain networks. In fact, there are 100 billion neurons in the human brain, but there's a 1,000-fold number of connections between neurons. So each neuron is connected on average to 1,000 others, and all of our brain function is determined by these connections. Symptoms localize to networks rather than any anatomical regions in the brain. To localize the symptoms, we have recently developed a powerful new technique termed lesion network mapping. Using this technique, we can map brain lesions that have caused specific symptoms to a so-called wiring diagram of the human brain, and use this to identify the circuits causally linked with the symptoms. We have already used this approach to a number of different neurological and psychiatric symptoms, identifying the key circuits in these symptoms. Our overall approach is comprehensive. So we start from brain lesions, we map the symptom-specific networks, and then use state-of-the-art neuroimaging tools to fully characterize what is going on in those networks and what is the key problem in the patient's brains. The next step is to use neuromodulation and modify these networks in order to improve the symptoms in these patients. And finally, if all of this is successful, we are ready to move to randomized controlled trials that are necessary to really translate these findings into new treatments. Our work also tells about the molecular level impairments in neurological and psychiatric patients, providing targets for pharmacotherapy. At the moment, we have more than 10 ongoing projects, ranging from lesions all the way to the neuromodulation trials. To mention a few, our brain study aims to be the world's largest prospective lesion network mapping study, characterizing over 100 neurological and psychiatric symptoms. The PSMD study is an international multi-center study aiming to collect the world's largest dataset on post-stroke movement disorders. And our current neuromodulation experiments include use of transcranial magnetic stimulation, deep brain stimulation, and MR-guided focused ultrasound in combination with MRI and PET imaging. Currently, there's a bit more than 30 people on board, and we put a high value on team member well-being and truly work as a team. And we're hoping to invite 1 or 2 postdocs through this program to join our team. So hope to see you in our beautiful hometown, Turku. Thank you.
Thank you very much, Juho. Next, we will hear from Sara. Please.
Yes. Hello, everybody. I am Sara Whitford. I'm an assistant professor of biotechnology from the biotechnology unit of the Department of Life Technologies. So my research group is developing in vitro diagnostic tests for cardiac biomarkers, which means that, I have to start from the beginning. Yes, I hope, yes, this is the correct slide. So we're developing laboratory tests that could be applied to the automated systems or point of care devices used at the hospital laboratories to help in diagnosing or predicting risk for cardiac diseases or acute cardiac events, such as myocardial infarction. The tests that we're developing are based on immunoassays, which means that there's always antibodies that will bind the target molecule. And to develop the tests, of course, we have to have understanding of the target molecule. What could be a new biomarker that could be used for improving diagnostics? Could it be, for example, a special form of the known biomarkers that are already in use, but some improvement is required? And when we have an idea of the molecule that has to be detected and in the test, so then we have to optimize, first of all, the combination of antibodies to be used in the test so that the antibodies will specifically detect the molecule or the special form that we want to measure. Also, we are usually looking for molecules that are present in very low concentrations in the samples. So this means that we need high sensitivity of the assays. And this also means that we have to use label technologies that provide very strong signal so that it can be seen by the devices. We are really lucky in that in our unit, there is a group of Professor Tero Soukka, and they have been active in developing novel, innovative label technologies, and we can utilize their labels to produce highly sensitive assays. We also have to optimize the assay conditions. For example, sometimes it's important to develop very rapid tests, so this has to be taken into account, and also to make sure that the assay works reliably. It is usually not that difficult to develop tests for detecting molecules in buffers, but actually when we bring in the patient samples, let's say human plasma or human serum, which contains more than 20,000 different proteins, things get problematic. So this means that we have to use a lot of time and effort to optimize the assay to work perfectly also with the patient samples, and also to overcome possible other interferences that can occur in immunoassays. And finally, we have to meticulously evaluate the analytical performance of the tests to make sure that they are performing reliably, producing reliable results at the concentration levels that we want to measure. To develop these tests, of course, we rely on the use of patient samples. We have to have access to patient samples, and especially at the point when we want to confirm the clinical utility of the tests. So this means that we are doing a lot of collaboration with clinicians and cardiologists. Luckily, we have here locally in Turku brilliant cardiologists at the University Hospital of Turku. And we are doing a lot of collaboration with them, but we are also collaborating with many universities and hospitals around Europe and in the US. And I want to give you quickly an example of our latest research. So nowadays, for diagnosing myocardial infarction, tests for cardiac troponins are widely used. But the problem with the current highly sensitive tests is that when a patient comes to the hospital with symptoms of myocardial infarction, actually 50% of those patients get elevated results with the tests, while only 10% of them are actually having myocardial infarction. So 40% of the patients are getting elevated results while they are not having infarction. The reasons usually, well, there are many reasons. For example, patients who have problems with renal function, or patients with atrial fibrillation, infections, or maybe just strenuous physical exercise can elevate troponin levels. So it would be better to have a test that would identify specifically the patients who are having myocardial infarction. And for that aim, we have developed a test that specifically detects only the long forms of cardiac troponin T. And with this test, our preliminary results show, well, first, I must say that with the current tests, these tests cannot differentiate between patients who are having problems with renal function and patients who have myocardial infarction. While with our approach, we can actually use the test and calculate the fraction of long troponin forms. And this way, we can really well separate these two groups. We can much better identify the infarction patients. And we really hope that in the future, this test could help improving the patient care and diagnostics of myocardial infarction patients. So we are currently looking for one candidate who has already wide knowledge of immunoassays and has extensive experience in conducting immunoassays and also developing sensitive immunoassays. Thank you.
Thank you very much, Saara. Our third speaker is Juhani Knuuti, and I'll just get your presentation from here. Okay, your turn, please.
Hello. Yes, I'm here presenting the three teams, which I think are interesting in the point of view of postdocs position. I'm actually representing a team which consists about 25 investigators and 6 postdocs already. And this is a joint effort by the Turku PET Center, focusing on imaging, and Heart Center in the University Hospital. We have access to practically all advanced cardiovascular imaging techniques, of course, the PET imaging, but also CT, MR, and echocardiography. And we have access now a huge databases jointly with our collaborators in Leiden, in Amsterdam, in Utrecht. I'm going to now show the 3 main themes, which I think should be benefiting of having a new postdoc on the board. So the one is the advanced imaging in coronary disease. So we have actually done a lot of research during the last few decades, which are focusing on the coronary CT angiography, but perfusion imaging and echocardiography, especially for the detection of non-obstructive and obstructive disease as well as outcome. And here we have also combined our databases during the last 15 years together with the Amsterdam database. So we have a good access to look details. And we are also applying AI here to analyze, especially on the CT angiography. The other one is a more recent one, which is the quantitative perfusion imaging with a total body PET. So it means that you can image whole body at the same time. So that allows us to really quantify in absolute terms the perfusion in any tissue cell in your body. And this is now an ongoing process. We have already recruited 300 patients for the trial, so it's prospective trial. But there's a lot of potential to do nice research, focusing, for example, the technology itself, the analysis platform, which is now already designed, but there is a lot of improvement possibilities related to AI analysis, but also the clinical findings, what we get. For example, now we are recruiting patients with the chest pain and suspected carotid disease, and we want to understand what happens to different organs, for example, brain, kidneys, and other organs, which are known to be potentially associated with the atherosclerosis. The third one is focusing on the molecular imaging, mainly on PET. We have very good access to various PET imaging tracers for different processes mentioned here: angiogenesis, inflammation, the new folate receptor beta is coming to human use for the inflammatory targets, and also fibrosis with the FAPI tracers as they are now coming. So these three projects for me represent a good chance for a new postdoc to learn a lot, but also we hope that the person who is coming is able to integrate the team, also bring his or her expertise, and hopefully has an access or experience already on the imaging related to all those three different topics. Thank you very much.
Thank you, Juhani. So our fourth presentation about these projects and research we are having here is by Matti Kaisti.
Hello. So my name is Matti Kaisti. I'm an assistant professor here at University of Turku, Department of Computing, and within the department, Digital Health Laboratory. So we are engineers and scientists and computer scientists. So we are a bit different, that we are not from a medical background. So just a few words about our sort of health technology lab. So basically what we work with is digital new novel health tech solutions and simply promoting digital health and well-being. So we have a fairly broad topics in our lab and in our department. All in all, in the department, it's one of the biggest ones in the University of Turku, close to 200 people, and our lab has around 30 people, and we have 3 professors, one full professor and 2 assistant professors. And there's also a link to our website for more information. So basically, we have three positions available here in our lab. I would be hosting one, and 2 of my colleagues would each host one. So this is a topic that I'm especially interested in. So 2 other topics are also available in the SYS-LIFE webpage. But all in all, the skill sets are very similar. And we are interested in AI methods and on wearable signals, sensors, and instruments. And this is one topic, but it's not necessarily if you are applying to work with us that it's exactly like this. But if it's broadly you're interested in AI health informatics or biomedical AI, this would be sufficient, and we would be happy to have a discussion if you have any questions, if we can find a common ground on the projects. But that being said, I think some of the topics that we are especially interested in is using new AI-driven monitoring techniques using body sensors and wearables. So that's some of the things we have been doing quite a bit, and we are interested especially in multimodal data. So we can get mechanical responses of the heart, electrical responses of the heart, and circulatory responses, and we can do this in a different type of settings. High-risk patients and screening, for example, one good example would be on this topic that is already a commercial topic, but would be the Apple Watch and atrial fibrillation. Of course, we are not doing that because it's already done, but are very similar type of solutions on a more difficult cardiovascular diseases. Then we are also interested in not only doing wearables and monitoring techniques, but new trustworthy models and methods. So lots of the time in AI health informatics, most research is I would say, untrustworthy, overfitted models on small data sets, and basically they are just aiming to create good predictive numbers, but we are more interested in that we can trust these numbers and creating new methods how to evaluate the trustworthiness, and creating models that can achieve it. So that would be one very interesting topic also to consider. And multimodality is something that the world is going forward to. So having different sensing modalities combined and seeing if those can improve the predictive performance of the models is quite interesting to us as well. And one a little bit more a bit different topic would be to incorporation of state-of-the-art machine learning models in a computationally constrained computing or microcontrollers. So basically, if we would take a big model that works very well offline, then how to use it at the point of care real time when you have to use a wearable system that doesn't have the same computational power. So how to achieve that would require some algorithms specifically tailored for some hardware. So what we can offer, so we are truly a multidisciplinary team, and we are very happy to collaborate with clinicians and doctors and get the support from that side. And if you would be applying for us, we would have more than one professor supervising you, if this is needed. So my expertise is in health informatics and instruments and monitoring solutions and machine learning, but we have some very good experts in machine learning and AI as well that can supervise you. We would like to give you a possibility, if this is what you like, to supervise also other PhD students and other junior researchers, or if you require to be trained towards your independent career more, to have more supervision yourself by us. And we are very happy to introduce to our international network and promote collaboration possibilities with other people as well. And we work quite closely as well with our clinical informatics, which is basically a data lake here from the southwest Finland. So depending on our interest, we can extract some of data from this data lake as well. Thank you.